Hello, everyone, and welcome. Thank you so much for joining us this evening to discuss an incredibly important topic, rare disease research and how stem cells are helping to unlock new treatments and understanding of these diseases. There's a huge unmet need to provide better options and treatments for people who are suffering from rare disease. The pharmaceutical industry has not traditionally invested in this area. The patient populations are low, and accordingly, the profits are low. And so these diseases typically have no treatments, and they're very challenging to study with small and dispersed patient populations. We have focused on rare disease since we began because stem cells offer a unique opportunity to actually create a model of these diseases in the cells themselves. And by doing that, try to tease out the root cause of the disease and understand how to develop treatments and therapies to address them. So rare disease research is also incredibly valuable for understanding more common diseases because the impact of very specific genetic features that many rare diseases have give us an understanding of the disease biology in general. You're going to hear more about how we do this from NYSCF's Dr. Dan Paull, who is working closely with our panelists and collaborators, Matt and his daughter Grace, and Leena and her daughter Aria. Leena, Matt, and their families are passionate, brave advocates and pioneers of research into the rare diseases that affect them and millions of other families. Their commitment to finding new options is truly humbling and inspirational, and we are so happy to have them with us today and to partner with them on this important research. So many of our projects in rare disease have been through partnerships with the foundations that incorporate the patient voice, which is critically important to getting this research right. Coordinating the research participation is not necessarily an easy thing to do. Funding, never easy. Collaboration with people being, treated with from, clinicians all over. And the need for the increase in advocacy efforts. The bottom line is that advances in rare disease equal advances everywhere. And rare disease patients and families need better options, and looking for those options is going to help us actually create not only options for those patients and their families, but for all of us. So on that note, I am absolutely thrilled to welcome our panelists today. And I would like to introduce our moderator, Dr. Raeka Aiyar, who is our Associate Vice President of Scientific Communications. We're very lucky because Raeka has an extensive background in genetics, has worked on rare disease research herself. So Raeka, why don't you tell us more about rare diseases and launch our conversation tonight? Thanks so much.
Thank you so much, Susan. I'm really honored to be moderating this panel, and we have a really outstanding lineup of panelists with great perspective on the unique challenges that rare disease presents, as Susan mentioned. So I just want to clarify what it is exactly that we mean by rare disease, for those of you who may not be aware. Rare diseases are conditions that affect less than 200,000 people in the United States individually, and there's similar definitions worldwide. There's an estimated 6,000 to 7,000 of these rare diseases in total, so they're actually not that rare. That's a bit of a misnomer. They affect something like 30 million people here in the United States. They are mostly genetic in cause. They're usually inherited, often caused by a single mutation, which makes them, very relevant for from a genetics perspective to study. And we'll hear more about that as our discussion progresses. Some rare diseases are not, and the cause is unknown. So there's a lot to uncover in this research. 70% of rare diseases or so are pediatric, meaning that, that children are affected at birth or in early infancy. And they often have very, very severe consequences, deadly consequences. So this is a really, really urgent unmet need, as Susan mentioned. And most importantly, these are generally orphan diseases, which means that there is no pharmaceutical research, there are no treatment options. And this is a really important thing to turn our attention to. So the lack of investment in these diseases means that there's very little knowledge and understanding of what causes them. And of course, that makes it very difficult to find therapeutic options. So what this ends up meaning is that parents end up taking things into their own hands. And we're going to hear from two such parents today. I can only imagine what they have gone through as parents. My heart goes out to them, and what they have done is truly inspirational, and we'll hear about their journeys momentarily. The good news for all of us is that stem cells and other exciting new technologies are offering amazing opportunities that we couldn't even have a discussion about several years ago. So that is really, really promising, and is making us all hopeful and gives us something to talk about today. So I invite all of you to submit questions via the Q&A function. We did have some audience questions that were submitted already, so I'll be incorporating those as we go. So thank you so much for those who submitted them. So I want to start by bringing on our panelists. I'd first like to welcome Matt Wilsey, the president and chairman of the Grace Science Foundation. Matt, thank you for being with us.
Thanks for having me.
Of course. You were a Silicon Valley entrepreneur, an angel investor, and a startup advisor turned a rare disease advocate. Your daughter Grace was diagnosed with a rare disease called Ngly1 Deficiency in 2013. And so you and your wife jumped into action, set out to take this disease on, starting the Grace Science Foundation in her name. You all have achieved such an extraordinary amount in just a few years. I've had the privilege of following your journey. And you've mobilized some amazing scientific experts worldwide to turn their attention to a disease that they probably never heard of before you guys came along. So could you talk a little bit about your journey as a parent, as an advocate, as a foundation chair, as a research coordinator, and so much more?
Yeah, absolutely. And you and Susan did a great job summing up rare diseases. That's probably about the best intro I've ever heard to kind of tee this up for folks that aren't familiar with it. But, I don't think Grace is 11 now. And if someone had told me I'm going to be doing this 11 years into the future, I would have said, you're crazy. But, really a parent, as many people probably listening, you do anything for your kids. And so the cards that Krista and I were dealt, we just knew that we had to mobilize, because we had people tell us, just go home and enjoy your daughter. And we also had doctors tell us that she had 5 years to live. That was her expected life span, given her phenotype, the way she presented in the clinic. And just given sort of my background as an entrepreneur, I didn't want to sit back and take that avenue, and thought we can do something about this. But unfortunately, the disease was not known when she was born. So we had to go through a very long, extensive diagnostic odyssey that a lot of patients go through. And most people don't even make it through that. A lot of these kids with rare diseases, those 6,000 to 7,000, are dying long before they are diagnosed. And if they are diagnosed, they just don't have the stamina to keep working on it. So, partners like NYSCF are super important to keep working with the parents and the advocates who are pushing forward. And there's a lot of hope in our world for Ngly1 deficiency now because of partnerships like this.
Really glad to hear that. And I think, as you said, this is such a common experience that parents go through, that, you'll have no idea what's going on, and the doctors have no idea. And you just have to spend so much time just on the diagnosis alone. I mean, thankfully, now genome sequencing has become more accessible. So when it comes to genetic diseases like Ngly1 deficiency, you can at least find a molecular cause, but that's of course not the whole story.
Exactly.
But,
exactly.
We'll talk more about that as well. I'd like to next welcome Leena Panwala, who is president of the INADcure Foundation. Leena, thank you as well for being with us here today.
Thank you for having me. And yeah, I agree with Matt. That was a great introduction to rare diseases. My journey is similar to Matt's and to probably most parents out there. We we started the INADcure Foundation after we got a diagnosis of this, disease that we had never heard of, that nobody had ever heard of. And so our daughter was 2 at the time. She's now 6.5. And, we felt compelled to become a voice for the disease, to really kind of bring some awareness to it, help advance the research. There wasn't, obviously funding is always an issue when it comes to rare diseases. And so there really wasn't much kind of information out there available to us. And so we really kind of scrambled to piece everything together that we could find to try to not just help our daughter, but to help other children affected. And, and that helped us kind of launch the foundation and really do what we could in our power to advance the research for a effective treatment, ultimately at the end of the day.
Yeah, absolutely. And I was going to say, I mean, you guys have done such an amazing job galvanizing all of this information from all these resources, getting it onto your website in a really structured way and helping to build that voice for the parents. And I was also going to say, you have a background in operations and management and also as an entrepreneur. So I think, I can imagine that that's served you well in this kind of coordination and, scientist role that you've had to step into effectively. So can you talk about that a little bit?
Yeah, so it's helped. I just dove right in. I had limited experience in the medical field and in the nonprofit world, so I kind of went in really headfirst and just learned a lot as I kind of went along. But I think that having the background in management kind of allows me to coordinate, with the researchers and really, try to have everybody work together to bring, advance the research, bring the research to a point where we can, get some useful data from it, that can ultimately help these children.
Absolutely. And Matt, I think your background as an entrepreneur has similarly helped you to be successful at some of this, I understand.
Yeah, I mean, absolutely. And I would say a lot of the other successful advocates in the rare disease space are entrepreneurs. And I would say that they're sort of the tip of the spear right now in this sort of new wave of medicine. And it really sort of screams for technology from academia, but CROs and partners like NYSCF that they can help build some of the infrastructure that makes this a little bit more turnkey for different disease groups. So it's more efficient, and with that efficiency lowers the price ultimately of therapies. So I think that the kind of the lessons from this early wave will apply for generations to come.
Oh, that's absolutely a great point. Yeah. I next want to welcome Dan Paull, our final panelist, who is the Senior Vice President of Discovery and Platform Development at NYSCF. Dan, obviously, aside from being an expert in stem cells and their role in disease research, you worked on many innovative methods to maximize the benefit of stem cells for research and their use in therapeutic development. So, because of that, for example, you've led the development of the NYSCF Global Stem Cell Array, our technology that allows this kind of stem cell disease research at scale, and led our rare disease research collaborations at NYSCF for many, many years. So could you tell us a little bit about what brought you into rare disease research, why it's important to you in the world of stem cell research at large, and kind of talk about how you and the team at NYSCF have decided which rare diseases to work on, or have they sort of chosen you? Or
Sure, yeah. I think that was a perfect segue from Matt as well, because that sort of underpins a lot of what we've tried to do here over the past decade plus is really develop platform technologies that can lower the barrier to entry for be it a common disease, or in this case, like a rare disease as well, which obviously equally as important. And so in reducing that barrier to entry, for us, that's meant applying like a level of standardization and a level of automation that kind of wasn't really out there, especially in the stem cell technology space before. And so what we've been able to do is as a team, bring together engineers, both software engineers and robotics engineers, data scientists, and obviously biologists, and have built this platform that's really sought to bring groups together. And so for the most part, it's been that these foundations like Leena and Matt's have reached out to us, and where we can then provide that help and support. And the way we've been doing that is by building, helping develop models of these different diseases. And so right from the outset of developing the platform, some of the early work we were doing was with a program called the Undiagnosed Disease Program, which is comes out of the NIH, where a number of patients who have clinical manifestations of different diseases and don't fall into any one given disease bucket. Right from the get-go, rare diseases was like a major theme in sort of the development of cell lines here. And so as that's progressed, we've been able to kind of harness the technologies that we've built and then work very closely with the clinical research team that we have to work with foundations like this and help them sort of across the board enable ways for researchers to get hold of different types of materials. So for us, it's been everything from helping to arrange collections of different samples to then making stem cell lines, and we can talk more about how we do that in a moment. But even more importantly, perhaps, is also not only internally using them, and we'll touch on that as well, I'm sure, but being able to make them accessible to the research community, because that's one of the big barriers as well, especially in rare disease, is just access to patient material. A researcher may well want to be working on a given disease, but just being able to obtain relevant samples from a given patient is incredibly difficult. Things can get held up with all sorts of paperwork and bureaucracy of trying to transfer samples around from institution to institution. So here we've made a really concerted effort to try and minimize that as much as we can, so that these samples can go out to the wider world. Basically, these are sending samples all over the world.
Yeah, absolutely. Thank you for setting that up. I think that's a good segue into talking now a little bit more about stem cells and the science behind that, and how we, why we think this is such a valuable opportunity. So I want to go back to our advocates now who came to us, and we're delighted to be working with both of you on this really important research. And I'd love to hear from each of you a little bit more about the science behind your daughter's disease, why you got excited about adding stem cell research to the kind of general portfolio of research that you've both been harnessing over the years, and kind of what brought you to NYSCF and about our work together. So Leena, maybe you want to start this time?
Sure. So, I connected with NYSCF when I started reading more about how powerful stem cells were to really helping treat and cure rare diseases. The knowledge that we can gain from looking at these cells, I thought was very kind of important to understanding the disease mechanisms, and again, ultimately coming up with a more effective treatment option. And so I really just didn't know how to get to that point where we had stem cells. And that's, I was fortunate to be able to connect with NYSCF, and I'm so grateful to have, an organization like yours available, so that we can make it easier for our families for sample collection. And from there, the collection can be turned into stem cells, and then, very effortlessly be transferred to, to research labs. And so we can take a closer look at those cells to get a better understanding of the mechanisms within the cells and what is actually taking place, and the effects of these diseases at the cellular level on these children. And so, I think that's kind of never been really done before, at least for our disease. And so this was, again, a whole new world for me, and I definitely learned a lot.
Yeah, and can you talk a little bit about what INAD is and the kind of the science behind it, and what the open questions are that you're trying to get at with stem cells?
So INAD stands for infantile neuroaxonal dystrophy, It's comparable, I guess, to Parkinson's, where you see a loss of gross and fine motor skills, as well as Alzheimer's, where you see a decline in cognitive function. So, you know, our children are born normal. They hit kind of those earlier milestones. And then, you know, after the first year of life, you start to see some regression take place, and then they just continue to kind of regress from there on. And so, you know, we just we just wanted to help kind of learn more about why this is happening, you know, how we can maybe stop it from progressing, stop the disease from progression, from progressing further. Yeah, and so that's kind of why we're, you know, pushing for this research to happen.
Great, yeah, of course. And Matt, could you tell us a little bit about Ngly1 deficiency and where you see stem cells fitting into the research that you're pushing as a foundation?
Absolutely. So Ngly1 it's ubiquitous. It's found in every system in the bodies. It's primarily in the central nervous system. That seems to be where our patients are most affected. But we see issues in muscle and peripheral neuropathy in the liver. So it's global. And there was one of the big reasons very early that we invested in stem cells in a variety of places. First at Stanford University, but then also at Gladstone in San Francisco. And we expanded and started to work with the team at the Salk Institute as well. And all were phenomenal, but we really felt like this was an area of science that we needed to put more chips on the table, so to speak. And we kind of wanted to see if different teams would come up with the same answer. And what our two main purposes, the first was to understand the pathway. Where is Ngly1 actually important and where is it not? We could do that much easier in iPS-derived cells. So which types of neurons are the most important? What leads to deterioration as the patient ages? And then once we have a finding, to hammer those cells with different therapeutics that we could actually leverage that as our platform. Because animals, while fantastic, do have the limitation of cost, and they might not also replicate the disease. And you could cure an animal, but you can't cure a human. So we really wanted to focus on those two initiatives. And all those aforementioned institutions did find very interesting things. And one of the reasons why we wanted to take this to NYSCF was we wanted to verify it. And then we wanted to expand at scale. And as Daniel mentioned, we also need to provide access in a very easy manner. Sometimes academic institutions aren't that easy to get cells out of, which we did experience, which is really no one's fault. It's just bureaucracies. So those were the big reasons to work with NYSCF, was to really verify, expand, and then distribute at scale.
I think that's a great segue into Dan's part. So Dan, if you could talk a little bit about, you know, some like how the idea of scale and stem cell models have helped us to look at rare diseases, what other types of rare diseases we've looked at, and some lessons we've learned through that work there.
Yeah, absolutely. So, and just to take a step back, just to put in context what we mean when we're talking about the types of stem cells here. So for us, everything starts with a pretty small sample, really. It could be a tube of blood, like going to the doctors and getting a blood draw, or it could be a small skin biopsy, maybe the size of an apple seed, small piece of skin. And from the cells in that, thanks to some incredible technology that won the Nobel Prize a few years ago, we can turn those samples back into stem cells. And from there, that's really the stepping stone to why this is so valuable, because of course, it's not really the stem cells that we care about so much as what they can turn into. And so you can kind of see over my shoulder a little bit, some pictures, and it's the fact that we can make brain cells, we can make different types of neurons, we can make liver cells or muscle cells, whichever it sort of needs to be. And that's sort of really been the driving force behind a lot of, behind really everything we've done here. And so for us, scale in that context is just having enough samples to be able to find a result that holds true and isn't just sort of an artifact of a one-off experiment or a particular sample. And so sort of in your background, Raeka, you can see some of the robots that we have here in the lab. And really the intention there is that instead of a typical scientist who might have maybe the samples of 3 or 5 patients at a given time, the capacity we've put in place here allows us to have, you know, hundreds in a plate at the same time. And so those experiments then allow you to do obviously either more drugs or a single drug across lots of different people at the same time. So, so from the get-go here, yeah, rare diseases, as I mentioned, has been sort of a major focus. We've worked with foundations from diseases like Batten disease. I mentioned the Undiagnosed Disease Program, foundation looking at Jordan syndrome, Charcot-Marie Tooth. It's really covered a whole spectrum, and it has been great to see these samples go out to different researchers and sort of research continue then outside of here. Internally here, what we've been really interested in is, I think one of the things that sort of has been touched on as well here is that these diseases are what we call monogenic. So they're often a single gene that's causing the problem. And that's great, but also can be a challenge as well. It's great because you kind of have a clue as to where you need to look for, but you don't necessarily have the benefit of knowing exactly how you're going to measure it. So if you have a neuron, what exactly are you looking for that's changed in that neuron? And so one of the things that we've been very interested in over the past few years is developing technologies, again, that can be somewhat agnostic to a disease, but we'll look at, you know, it could be Ngly1 or it could be INAD, and maybe we can apply the same sorts of assays and same experiments to look at how the cells are changing, and then when we have a drug, how that changes it again. So that's sort of really been the driving force behind a lot of what we've done here, leveraging, of course, all the other work that we're doing in the background that allows us to learn how to make oligodendrocytes like you find in the brain, or microglia, or cardiac cells. Sort of this all-encompassing approach that we've got here really allows us to then straddle across all these different disease areas.
Right, all these different cell types that are key for the functions that we're looking at, because
Exactly.
It's often not just one cell type that's implicated, but many, as Matt mentioned in the case of Ngly1.
Precisely.
Could you talk about how CRISPR gene editing has been useful for stem cell research in rare diseases as well?
Yeah, for sure. So, so again, come back to that concept of this being, you know, I think one of the things with rare diseases is that it's gotten to the point where I think it's the barrier to understanding which gene has changed is much easier now. Sequencing has become sort of almost a standard of care to some extent in terms of diagnosis at this point. And that's great because you can learn relatively quickly maybe what is different in that person's genome. But again, what the implication is sort of unknown. So if we can grow the cells that have this unique change about them, we can use something called CRISPR, which is a tool that allows us to repair that change. So if something has changed that shouldn't have in the DNA, we can change that back to sort of how it should look normally. And so that tool allows us to have then sort of make both cell types. We can have the cells that have the DNA where it's incorrect and the DNA where it has been corrected, and see how they're behaving slightly differently. And that's kind of the perfect control in science. You can't really get much better than that. And then, of course, being able to do that across multiple people allows the power of the study to get stronger and stronger. So for us, it's become an invaluable tool. I think the next step beyond that is, you know, that's great for us and we're maybe more interested in sort of therapeutics and drug screening and that approach. But the clinical application of that is also obviously very apparent in terms of if you can grow the cells that someone that are defective, but be able to repair them, could you then transplant those back in? And so we're seeing that start to happen in all sorts of different diseases. We just saw an IND be approved for Parkinson's disease in that space where...
Could you say what an IND is?
Oh, sorry, yeah, sorry. So in order to be able to take a point, in order to be able to sort of perform a therapy in people, you first obviously have to get approval to be able to do this. And so that approval was just given to begin that in Parkinson's disease, injecting the specific type of cell that's sort of defective in Parkinson's disease can be now begin experiments to inject those back in. So, you know, that holds tremendous implications for these diseases where similar sorts of loss of cells or disruptions in the way the cells are behaving, you know, it's sort of within grasping distance of maybe a therapy that can one day be applied to these rare diseases as well.
Right, absolutely. So that's really important, and we'll get a little bit more into the therapeutic options in a moment. I also wanted to talk, you know, sort of while we're on the science of what's going on here, and the things that we're learning from rare disease about these rare diseases. I think all of you are pretty familiar with the implications of rare disease research for a variety of other diseases, and that you've seen come up as you've sort of go along. Could you speak to that a little bit? I think that's you know, it's obviously a tool that's really helpful in advocacy to helping people understand the importance of it, but you know that investing in this research can have so many implications for diseases that millions of additional people suffer from.
Yeah, again, you know, I think INAD is a great example. Leena touched on the fact that it sort of shares these hallmarks of Parkinson's disease is one example here. And so we're in studying this disease, we're sort of understanding now what goes on inside the cells. And what we're beginning to see, and not just us, but this is sort of building as more people get hold of this material and from studies that have come before, is we understand that the way a cell behaves and the way we see it sort of get disrupted actually has very similar disruptions in something like adult Parkinsonism. And so if we can identify a therapy that seems to protect these cells or helps them function more normally, perhaps, you know, it gives you an insight into maybe that's going to be just as beneficial in a disease like Parkinson's. I think this is also sort of one of the problems of not knowing necessarily in more complex diseases like Parkinson's or Alzheimer's, where there isn't a true single gene that's maybe causing someone to develop that disease. You're probably seeing the cumulative effects of lots of different things, and it could be that just interrupting those signals at one point, and it might share the hallmark of one of like a rare disease, is enough to sort of provide some level of benefit in that disease as well. So anything that helps us gain an understanding of biology, there's still so much we just simply don't understand, is beneficial, and these rare diseases really are a great insight into understanding that.
Right, because often when you find the gene that's responsible, there isn't a whole lot of information about what that gene even does. So this is a great opportunity to find out why it's so important for the function of cells, right?
Exactly.
Leena and Matt, anything to add to that?
Yeah, go ahead.
And I just, you know, just kind of touch on what Daniel said. The stem cells, I think, can also offer more kind of precise, you know, treatment options based on one's own specific mutation. At least for INAD, all of our children have different mutations on the PLA2G6 gene, but every child has a different mutation. The symptoms are, you know, typically similar, but, you know, to be able to effectively treat, you know, our children's particular mutation, I think may end up being a much more promising, may have a much more promising outcome, for sure.
The only thing I would probably add, Raeka, is that kind of to Daniel's point, an example, when Grace was 1 or under 1, if I talked to 100 donors / investors or C-level executives at large pharmaceutical companies, and I said, rare diseases unlock common diseases, I think I would probably only get maybe 5 that agreed with it. If I asked that the same 100 people today, it would be 100 out of 100. It wouldn't even be a debate that rare diseases kind of are a gateway in how to treat some of these other diseases that we've honestly spent billions or trillions of dollars trying to find links or ways to treat. And if we can disrupt or fix one problem, that could solve a significant you know, characteristic of a much more common disease like Parkinson's.
That's really promising to hear, and I think that's a testament to the power of the advocacy work groups like yours have done over the years, and that's really, really great. I mean, and to the point of advocacy, I wanted to ask the two of you, Matt and Leena, what do you find that people most frequently misunderstand about rare disease or get wrong, and how, and maybe hopefully that's changed over the years, but
yeah, we get it a lot. And Susan covered a bunch of these. One, that it doesn't affect very many people, but 30 million Americans is a huge chunk of people.
Yeah.
And a lot of these people are just floating through the system. They're misdiagnosed, or they're diagnosed with something that provides services for them, but really no hope or answers. And I guess another one is that it's a waste of time and resources, but just like we were talking about, if it can unlock something more common, that's really valuable. I obviously leave no patient behind. Every patient deserves to be treated. And I think that, COVID has kind of opened people's eyes to this, because maybe it didn't touch them previously, or at least not directly. And everyone's kind of experiencing what a lot of rare disease families have experienced for years. And then I think the third one is that rare diseases aren't a viable business model. And we're starting to see companies emerge that are actually making this a true business. Companies like Amicus or Ultragenyx, Audentis, Bridge Bio. There's a whole host of them. Takeda acquired Shire, which it was focused on rare diseases. So we need more of that. And I think some of it comes with incentives, which hopefully we'll talk about later in the panel. But that's my greatest hope, is that companies start realizing that this is a business opportunity where they can sort of have that double bottom line. They can help people, but also make a profit for their investors, which I know is important for the system to work.
Absolutely.
I think there's also sort of I'm sorry. There's also a flip side there as well, where it's a unique opportunity. In the same way, I think one of the things that we've become very interested in is sort of the flip side to that, where there's all these drugs that already exist, and it was designed for one indication in one area, but may well have a benefit in a rare disease. And so again, it's sort of having these models now, reading recently about cystic fibrosis and sort of their quest to find a drug for treating that, or multiple drugs, sort of the agonizing over getting access again to patient material so they could actually test whether the things they were finding would really have an effect. The fact now that we can sort of, with this, I've said platform a few times, with this platform that allows us to go right from a patient's skin sample, blood sample, to having some neurons in a dish, all within the same place. What it's allowing us to do then is take drugs that have already been approved, medicines that have already been approved, and put them back on top of these cells with these particular genetic changes and on the right types of cells, and see if there's already a medicine that's out there that we just don't know would have an effect. And so that's sort of the flip side as well of this, is as much finding new things as about repurposing. And again, we shouldn't forget that these are, as you said earlier, orphan diseases. There is just no treatment, and predominantly it's children that are affected with this in a lot of diseases. So I'm sorry, Leena, I didn't mean to interject that.
No, actually, just before you jump on, there was a question about platform technology. I just wanted to make sure everybody understood what that meant before we keep talking about it. Dan, if you could clarify very briefly.
Yeah, for us, we think of platform as sort of this end-to-end, all-in-one solution to this effectively. So it has been that we've built the entire capabilities to go from, it even starts before having the sample. It goes from having the ability to help collect samples, as I mentioned at the beginning, through to turning them into stem cells, through to then doing a drug screen on the cell type that we're interested in, and having the right analysis tools, the right, it's everything. It's having the right software developers have built the platforms to capture the data so that it can be analyzed. For us, that's how we think of this, as a platform for unlocking therapies for these sorts of diseases.
For any disease it could be applied to. Yeah, right. Okay, Leena, our apologies. Please, please talk about the misunderstandings that you've encountered as well.
I think that even for myself, I just kind of naturally assumed that there was research happening or some sort of treatment option available. Once we were handed this diagnosis, my jaw dropped that there was really nothing out there. There was no information out there. So I kind of assume that there has to be something like there. I'm missing it. You know, there has
Yeah.
to be like it has to be there. You know, that explains why there's, you know, less than 5% of rare diseases with treatment options, because there's
Right.
really, yeah, there's really no research unless you have organizations like ours, patient foundations, really pushing that research forward. Because otherwise, you know, obviously things start slipping through the cracks. And, you know, with 7,000 rare diseases, you know, that's a lot to tackle, you know?
No, absolutely.
Yeah.
And that's, you know, I mean, you think like, okay, well, we've done this human genome project. We've sequenced all of the genes that humans have. Like, now that you know the gene is, why can't you do anything about it? And that is a definite, you know, sort of additional barrier. I worked on some of that back in the day, and there was this frustration in the public once they realized, like, oh, we haven't solved all of the diseases yet. We don't know what they're doing. Just because we know what the sequence of a human genome is, you have to now figure out, okay, how does that play out in the cell? Like Dan was describing, you have to, you know, CRISPR edit it. You have to, you know, do all of these different experiments to see how the cell is functioning or dysfunctioning. And all of that is really, really critical to go on with. And that's why, you know, it's the rare diseases are giving an opportunity to discover new features of these genes that we didn't know. But yeah, it's got to be so frustrating that we haven't figured it out yet, because it's been so many years since we did the genome. But yeah, thank you guys for elaborating on that. So I did say we wanted to get back to treatments. I think this is a good time to revisit that topic, because that's of course the ultimate goal of what we're all doing here. We're trying to find what are the opportunities for treatments. So Matt and Leena, I'd love to hear from you a bit more about what you think the most promising opportunities are right now for treating these diseases, and also, you know, the rare diseases in general. You know, where you see technologies, emerging technologies like stem cells, like the drug screening type things that Dan mentioned, like antisense oligonucleotide therapies, which we can explain when we get into that, gene therapies, all of the above. So anybody want to start?
I'm happy to jump in. I would say that we're trying all of them. We're trying gene therapy, we're trying ASOs, we're trying small molecule inhibitors, small molecule potentiators. We've kicked the tires on enzyme replacement therapy, which is a little bit tricky in the case of Ngly1. We're not trying to close any doors. As long as we can fund these different projects, we're going to keep pursuing them, because one therapy might work for some of our patients, but not for all of them. And we're not going to stop until we have a therapy for everybody. I would say the thing that I'm most excited about, and well, I guess ASOs are kind of there's sort of the shiny object. And
could you explain what they are a little bit?
You'll probably you and Daniel will do a much better job.
No, I'm talking to three scientists here. You explain that.
Daniel, why don't you take the ASO and then I'll hop back in.
Yeah, so effectively an antisense oligo is, if you think of DNA, if you think of DNA as sort of, we think of DNA as like a double-stranded helix, I guess. And so when you make it gets turned into RNA, and so you can sort of inhibit these things by having something stick to it, and it can either cause something more to be created or something more to be stopped. And so that's effectively what these sorts of technologies are doing. I think, you know, it's not quite the same, but one example of this is something like the Moderna vaccine in the COVID vaccines right now, which is these single-stranded pieces of, in this case, it's going to be RNA, but can be injected into the body. They enter the cells and then they do something. These kinds of technologies that directly interfere with either DNA or RNA are really opening up sort of a new landscape of modulating how the cells then can behave a little bit. So that's sort of all encompassing of these types of technologies.
Right. So with the ASO specifically, you're basically quenching the effect of the mutation that causes the disease.
Or something else downstream that is that's disrupting the balance of the equation. But and a lot of people in the rare disease world are pursuing ASOs. I still believe that gene therapy is the best approach for the vast majority of the rare diseases, and probably a fair amount of the common diseases as well, once we understand the underlying genetic reasons why the diseases progress later in life. I would probably draw the analogy in baseball that we're probably in the first inning still of gene therapy, just sort of the first wave of diseases. And obviously this is a field that's exploding, but there's not very many AAV types or lentivirus that we're using to target either specific cells. Science has not figured out redosing of these individuals, so it's a major limitation of the therapy. You can only give it once and then you're done. We have no idea how long it lasts, the durability. But I do think that gene therapy, basically gene is missing, replace that gene. And gene therapy is probably the best way to do that. Right.
And gene therapy you would be using to correct the disease-causing mutation. And
basically you put back the whole copy of it. It's not CRISPR where you're making a very precise incision and letting it repair itself. It's like the whole cassette.
Right. You're putting in the whole gene, a functional copy of the whole gene that's kind of malfunctioning in the disease. And you use a lentivirus to kind of achieve that effect, but there's a few different ways
or AAV,
AAV.
Or an AAV,
adeno associated virus.
Associated virus, yeah. Leena, how about you? What are you most excited about for the treatment prospects?
Yeah, I mean, I'd have to agree with Matt. We're not closing any doors. We're trying the small molecule compounds. We've actually recently successfully gene edited an iPSC line. I'm sure Daniel was involved in that. You know, we are excited about that work as well. It's cutting edge for us. You know, it may be further away than gene therapy is, but you know, we're hopeful that with the gene editing in place, we can see more of a kind of like a regeneration sort of neurons, you know, instead of just, you know, potentially regaining skills. If you can kind of catch the disease early on, perhaps, you know, these children can regain some of those lost skills. You know, and with gene therapy, you know, we hear it often, the earlier you treat, the better. But we just don't know, because it's never really been tried for our disease, what would happen if you treat once the symptoms actually are in place. So we're exploring all options, and I think it's important to keep pushing kind of all of them forward.
For sure. And I have a question for the two of you. You know, gene editing and using that in terms of being actually preventative of diseases is something that gets discussed often. Like, could you know, what if you could prevent having a child that would have to suffer from this disease? There's ethical discussions about whether that's the right thing to do or not. How do you two feel about that?
It probably depends on the severity of the disease. And I know, you know, some rare disease advocates would not do that. I think the future is that we will be doing in utero correction of babies. It will figure out what the disease is before they're even born. And we will be able to correct that. Now, the question is at what time, especially in the case of Ngly1, it's ubiquitous. It's in every cell of the body. So you got to do it pretty darn early to have that correction take place. But, you know, some people would argue we'll just have all expecting parents or couples, you know, do whole genome sequencing, and you just do IVF and eliminate that disease completely. The problem is a lot of these are de novo, they're spontaneous. You can't predict that they're actually going to happen until the egg and sperm meet and they are in the womb. That's basically where a lot of these are happening. So anyway, I think that in utero correction is definitely going to happen.
Leena, what about you?
Yeah, I mean, I'd have to agree. I think we, you know, could get to a place where we do see in utero correction. I'm also hopeful that we get to a place where we, you know, we have these genetic panels, and at least for our disease, you know, both parents are carriers. So, you know, what our families experience can be prevented, actually. You know, if you're aware that you're a carrier. So, you know, the preconception panels, I think, should be expanded to whole exome or whole genome sequencing. And so you have a very good idea of, you know, what your risks are when you think of starting a family. So
yeah, it's a different perspective when you've seen the sort of life-threatening effects that these mutations can have firsthand. So Dan, I wanted to ask you as well, in terms of treatments, what do you think are the most exciting avenues that we're able to provide to pursue at NYSCF? And you know, you talked about this platform technology that we have, and could you talk about, you know, is that something that a lot of people are using, or is it you know, something that we that gives us sort of a unique approach to things, and what other people might be doing instead that we could take advantage of?
Yeah. So you know, it's you know, major pharmaceutical companies are obviously very interested in drug screening and finding new medicines and things like that. And so the concepts of using robotics, you know, have been around for a while now. I think what we have done that is unique and proprietary to NYSCF is apply this to the point where, again, it's for us, we think about scale as having as many different individuals as we can in a dish. And so instead of looking for a drug that, you know, traditionally it's been done on a rat cell or some cell that is human but has all sorts of strange characteristics about it, we can actually use these cells that have come from people. We know everything about them. We know their clinical information. We know their genetic information. And we can do this across hundreds at a time. That is unique to us at this point, and really gives us this edge in trying to understand, both for a rare disease or for a common disease, that how genetic variation, Leena touched on it, even in a rare disease, when you talk about a single rare disease, it's not the case often that there is just a single change in the DNA that's occurred. And so being able to see if a drug that you think you've identified is going to affect 1 or maybe 10 individuals from a rare disease gives you such a greater confidence in whether you found a therapy that's going to affect the wider population of that disease. You know, talking about the ethical side, you know, as at NYSCF, we've done incredible things over the past 10 years where some of the rare diseases we were thinking about early on were in sort of mitochondrial diseases. And there we sort of pioneered some approaches to preventing mitochondrial disease inheritance. And so have thought long and hard, like from the inception of NYSCF, about like the ethical implications of many approaches for this. But I think where we are now with being able to use these iPS cells, these induced stem cells from skin and blood samples, and in particular for us, you know, whether that's a drug screen or something that looks a bit like a drug screen, I think is just sort of opening up all sorts of new areas, and we're quite excited about where that's headed, and hopeful, of course, that we can actually identify something that's going to translate into a treatment.
Right, absolutely. Could you talk a little bit about the prospects for stem cell treatments of rare diseases, what we know there, and what we might be able to do in cases like Ngly1 and Inet?
Yeah, again, I think it's sort of hopefully riding on the coattails of some of the other things that have come before us. And so these are obviously not it's not inexpensive to try and do a gene therapy trial or to try and do a stem cell transplant or something that might come down the road. And so what you're really hoping for, although maybe is not the case because bureaucracy happens everywhere, we were just chatting about this yesterday, is that still, even though it's incredibly rare, I think Matt can speak to this a little bit, that they're still being asked for just enormous amounts of additional experiments or paperwork or proof of different things, that it's not sufficient just to ride on the coattails of something very, very similar that's come before it. And when you are dealing with diseases where, you know, for some of these children won't live beyond, you know, their early teens maybe. And so how do you even begin to do some of the studies that might want long-term effects and things? I think it's very challenging, and it's probably going to require a little bit of a shift in mentality, but you know, I think the proof is coming that these kinds of therapies can work. It's just going to be how we get them to these underserved diseases and things like that.
I think that's excellent points, and a great segue into the final question that we have time for today, which is, you know, that you've all described some of these formidable challenges that you've had to overcome in the path against, trying to find rare disease treatments. What would you like to see happen in the next little while that could accelerate this path, make it easier for parents like you who come later? Are there lessons that we can learn from what you've gone through and what we've gone through in this pandemic with, having to go warp speed on the vaccine and accelerate approvals, accelerate treatments? Are there things there that we might be able to learn from to better serve these patients who don't have much time? Dan, do you want to start?
Yeah, I can start. I hope it's that in a couple of ways we can do it. One is, as I mentioned earlier, is sort of, can we repurpose existing medicines? I think that's one approach that again, if we can do it on the right cell types, I think that's where it's sort of maybe been hampered a little bit before, is that you're not always using cell types. So if there is a medicine out there that we can look to see if would have a benefit, I think it's also looking outside the box. I think people can think, oh, well, it's this mutation in this particular gene, and so there's no rationale for why medicine X or Y would work. But maybe we just need to be trying these, right? And actually just taking an approach and having good enough ways to understand if they are having an effect. It's going to require things like machine learning, potentially. It's going to require all these new technologies that we were putting in place here and have put in place to really understand that. I think it's hopefully it's going to happen because incrementally these discrete islands that existed of different disease foundations not being able to do certain things on their own, hopefully it's coming together enough that we can tackle it, and it's going to get easier and quicker. That's sort of the hope.
Absolutely. Leena, would you like to pick up on that?
Yeah, I mean, that sounds great. that would be awesome. Yeah, no, I mean, we're hopeful for that too. And just the research, the discovery phase, all of it is very, very complex, very time consuming. Obviously, funding is always an issue. And bringing things to clinic then becomes, even more time consuming, right? Like, you have to have the data in place. You have to have the natural history studies. You have to have, the criteria to bring something to clinic is, so large that, these, the diseases just to have an effective treatment out there takes a long time to kind of make available to the public. And so, if we can kind of cut back on maybe some of that criteria, or especially for rare diseases, right? Because time isn't on, their side. Time isn't on these rare disease patients' side. So, that would be, I guess, the ideal kind of world for people in my boat, I think.
Yeah, absolutely. Matt, you get the final word.
Yeah, well, I have like a whole laundry list of things that I'd love to see done that I think about basically every day and I add to them. But if I had to pick probably one, like drawing the analogy or the comparison to Operation Warp Speed is really an overhaul with the FDA of how these therapies for rare diseases are evaluated and vetted, which they have a very important job to do. And they've been absolutely wonderful with us. It's very tedious, and certain things they can't share with us from other diseases, which obviously they're bound by confidentiality. But so in a lot of cases, we have to reinvent the wheel, which takes, hundreds of thousands or millions of dollars in years that we really don't have. And so there's direct lessons from Operation Warp Speed that the FDA can apply to rare disease groups that come with a potential therapy that's really building on the back of another therapy that's already in humans. So it's not like we're starting from scratch. And so that's really my top priority, I would say.
Awesome. Well, I want to thank all of you so much for this discussion. I hope that I hope that we have convinced everybody out there why this is such urgent and important work, both urgent and important. And that we, anything we can do to help these amazing parent advocates as they lead the fight forward. We're definitely willing to do at NYSCF, and I hope all of you are too. So thank you all so much, and wish everyone good safety, health, and wellness, and hope to see you again at a future NYSCF event. Thank you all.
Thank you.
Bye.
Bye-bye.